Hello and welcome to another episode of Odds and Ends Season 3. In this episode, we'll be talking about the history of model organisms. Much of the knowledge of genetics and biology is based on model organisms, living things that share commonality with many other things. Within several different species, there are several overlapping features that need to be maintained in order for life to function within broad groups and families. Different organisms have been established, genetic simplicity, high generation turnover rate, and the ability to multiply very quickly. And we'll start off with Drosophila. Mendel's work was rediscovered in 1900 by Hugo de Vries and Carol Correns. In order to explain this in terms of genetics, Thomas Hunt Morgan chose a common fruit fly to do different experiments, and in the 70s and 80s, these experiments were built upon by Ed Lewis, who was interested in embryo development and genetics and molecular biology. To that end, Dr. Lewis created many different mutations in fruit fly populations that caused dramatic transformations in the body plan to develop a model of how animals grow and develop over time. With all the mutations gathered, a hierarchical network of how the genes interact with each other to create the body plan was created, and it was found that many of these genes are conserved in vertebrates, which is to say animals with backbones. And these studies became important for understanding how embryos develop and how disease develops in terms of birth defects. Now onto Neurospora. During the 1930s, due to the complexity of Drosophila, from a perspective of understanding cellular processes, researchers attempted to find more simpler life forms in order to study the cell on a smaller scale. The researchers George Beagle and Edward Tatum chose a single-cell bread mold called Neurospora crassa. It was chosen for its many benefits. For one thing, it has a rapid generation turnover. Number 2 is haploid in nature, meaning that instead of having 2 pairs of chromosomes like animals do, it only has 1 pair of chromosomes. This means that the effect of dominant and recessive genes can be immediately seen, allowing for quicker analysis of genetic recombination effects. Once mutations are made, different strains were developed that led to failures at different parts of the normal biochemical pathways within the cell's metabolism. This led to the discovery of the one gene, one enzyme hypothesis that stated that genes code for specific proteins, and for the most part, this hypothesis is correct in regards to how genetics works. To this day, Neospora is so popular to study metabolism, as well as gene regulation, chromosome behavior, DNA repair, DNA methylation or epigenetics, and other epigenetic related phenomena, as well as photobiology and circadian rhythms within plant cells. Dr. Joshua Lamberg, inspired by various other researchers, began using E. coli to study how E. coli reproduce both asexually and sexually, and through his information was able to codify the reality that genetics are controlled by DNA, and DNA is a source of heredity. This work proved that bacteria can reproduce sexually as well as asexually. Due to the greater ease of mutation when it comes to bacteria, as well as stabilizing these mutations, E. coli continued to be used as a model organism to learn about the structure of DNA. Indeed, the first known DNA polymerase was isolated from E. coli. Most DNA structure models are based in E. coli to this day. Now on to phages. Viruses that are specialized in infecting bacteria are called phages. They are studied under initiative of Max Delbruck in the 1940s. These species were studied to figure out how viruses replicate. They were also put into positions where they would infect different bacteria. It was through this process that was discovered that mutations for resistance are actually random, and those mutations become selected by virtue of the fact that they survive. Prior to that, it was believed that resistance to antibiotics and other stress factors were derived entirely from the selection process, but it turned out it wasn't the case. It turns out that mutations happen randomly, and some mutations are more able to survive in particular environments than others. Mutation creates variability, and the environment selects for the variability that can survive in such an environment. To this day, phages are still being studied in relation to other viruses to learn how they work. Next we'll cover zebrafish. In the 1960s, George Streisinger attempted to combine the current genetic screening technology of that era with vertebrate animals Through this process, he obtained a great many mutations that cover a great deal of embryo development. He also discovered that genetic duplication, something that's absent in mammals, indicating that genes within zebrafish are controlled by many redundant genes. But through similar genetic approaches, researchers were able to develop different models of how embryos develop and how the body heals. Amphibians and chickens were also used as model animals, especially in Germany. These animals were used to develop the mosaic theory of development, which stated that the cells determine the fate of the organism very early on. An idea that was disproved by the appearance of sea urchins which could regenerate their bodies. These models were not used very well due to the great complexity of these animals back in that time period. But it does illustrate the interest in using model organisms at that time. And these experiments led to the discovery of another model organism, the mouse. The mouse shares many different diseases of genetic origin with humans, such as cancer, atherosclerosis, hypertension, diabetes, osteoporosis, and glaucoma. Other diseases can be induced through genetic modifications like inbreeding. Such examples include Alzheimer's and cystic fibrosis. These mutant varieties have been used extensively to test out different pharmaceuticals to see which ones will work on people. They're also used to study how the genetics of different diseases manifest in the human body, and a genetic map has been made to see what type of genetic mutations cause different diseases. And to this day, slowly but surely, a full genetic map of all human diseases is being crafted. On a plant perspective, rice have been used as a genetic model for quite some time. The extensive research allowed for the discovery of 350 genes in 1935. Hybrid vigor and F1 hybrids were also discovered through the experiments of corn. Rice was also used extensively as a model organism due to its relatively small genome and similarity to other cereals in terms of morphology. This became a model organism for other crops such as wheat, barley, and oats, to see how the genetics play out in different systems. Ultimately, these two crops were replaced in their position as model organism by Arabidopsis. This occurred in the 1980s, and served very good. The species genome was relatively small, even smaller than rice. Its life cycle is very short. It can be genetically modified very easily, both through mutation and through traditional genetic engineering methods. And through the study of these organisms, the understanding of how stem cells work in the plant organ development, genetic pattern formation, how quantum immunity works within plants, how circadian rhythm works in plants, how much natural variation can develop over time, and how plants develop, have all been discovered using the Arabidopsis. Currently, Arabidopsis is the primary model of how plants work, and most of our understanding of plants is derived from Arabidopsis. We'll conclude by talking about pathogens. Tuberculous has been studied extensively for how antibiotic resistance is developed, and through the research it was found that antibiotic resistance is derived from mutations in genes that produce other proteins not used in resistance but are used in our protein formation processes, indicating that resistance proteins have developed from redundant genes used for the production of other proteins unrelated to resisting stresses. And the anticipation of these novel mechanisms is currently being developed to create a model of how resistance will happen and how new antibiotics will have to be developed for the future. Basically, this species has been used to predict what the antibiotic-resistant strains of the future will look like and what antibiotics will be used to kill it. And that book covers everything. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.